the introduction, and thanks for the organizers for inviting me to speak today. Also say hello to everybody out in Iowa. You can't see me in person. If you're not familiar with the Veterinary Diagnostic Center, we're a service center on East Campus, and we facilitate diagnostic testing for the veterinarians throughout the state and the region. A significant number of our caseloads cattle, so we do have some research projects going in diagnostic areas for cattle pathogens. So my email address and phone number are there. If you or your veterinarians have any questions after the talk, I'd be happy to follow up on any of those you might have. Okay, so today I'm going to talk a little bit about bovine respiratory disease, specifically the bacterial components of those. We're going to talk about different antimicrobials that you may use to treat BRD, some of the resistance mechanisms. For the last 20 years or so, there's been interest in emerging resistance in a lot of the enteric pathogens like E. coli and Salmonella. I'm going to talk about what we're seeing as far as resistance in the BRD pathogens like Mannheimia and Pasteurella. So we'll show some of that data and the trends that we might have going through the lab and what those look like. So bovine respiratory disease, it's the most costly disease of beef cattle in North America. Some of the studies attribute 70% of the mortality and others 57%. So that's a significant contributor to the mortality in the yard. In some studies, it's about 12.5 deaths per 1,000 cattle. But it's also insidious, so a lot of these BRD cases are getting missed. Some studies shown that 68% of those untreated steers had evidence of BRD, but there's no pulls and no treatments on those. So there are some that kind of go through without being noticed. It is costly. So this is a study from 2002 showed that there's $40.64 per calf getting one treatment. And then if you have to go to 3, that approaches $300. And that's 10-year-old data, so those numbers are significantly higher now with the cost of production and the price of those animals. Bovine respiratory disease is a multifactorial disease. So if you look here, this is what's called the epidemiological triad. So we have the host, the pathogen, and the environment are all contributing to that disease outbreak. So with the host, you need one that's susceptible. They usually have some immune suppression from shipping and then subsequent commingling at the yards. Some stress from that shipping and commingling. Then it's going to depend on the preconditioning status, vaccine status of those animals, what they were treated with at the ranches before they come in. Then the sources, is this a high-risk source or a preconditioned source? The environment, that's going to be, your humidity, your dust, your temperatures, your mud. All those are going to provide increase the stress on the animal. And then another thing to think about is the herd immunity. So is this a group of all preconditioned calves that has a high level of antibody to the respiratory viruses, or are they naive to those? Is there persistently infected calves? So there may be 1 or 2 animals that were persistently infected with BVD. They're going to shed a tremendous amount of virus and expose those to their pen mates and put them at higher risk for BRD. And then the pathogens themselves are these concurrent prior viral infections make them more susceptible to BRD, and new strains or serotypes of viruses and bacteria, and then are the vaccines appropriate in covering those different pathogens. So the first factor in BRD is usually going to be associated with a virus of some sort. Viruses are metabolic parasites that cause cell death or dysfunction in the lung parenchyma of those calves. So they're going to be have some compromised tissue. You can see right here, this is an IBR case we had submitted to the Veterinary Diagnostic Center. You can see it's got it's not a real healthy looking trachea there. That's caused by the virus causing some exudate, and this animal is going to be predisposed to a bacterial infection after it recovers. So they're going to cause this death and dysfunction of both the immune cells and the cilia of the trachea that are going to help reduce the bacteria that can colonize the lung. And then once that happens, the bacteria can set up shop. They're going to multiply rapidly, so within 24 hours you're going to go from 1 to 1 million or 1 billion organisms. And then they're going to start secreting toxins, which are going to compromise that lung tissue further. So this is, in the timeline of this, usually have 2 to 3 days for a viral infection, and then those bacteria are going to be 3 to 6 days behind when you're going to see the real fevers and sickness start to occur. Our main culprit here is going to be Mannheimia hemolytica. This used to be called Pasteurella hemolytica, but us as microbiologists like to change the names every year or two so we can stay employed. So this is going to be, now this is known as Mannheimia. This is our most prevalent isolation in cases of BRD in the diagnostic center. You can see here's a case of Mannheimia BRD. It's got what's called a fibrinous bronchopneumonia, so all this fibrin or exudate on top of the lung, this sticky stuff, that's going to be secreted by the immune system trying to deal with that infection. It secretes what's called a leukotoxin, so leuko means white. That's going to target white blood cells and try to prevent them from getting into the lung and stopping this infection. It also makes endotoxin, which is going to cause that fever, that sickness behavior that you'll see in these sick calves. Most of these are serotype A1, so that's a way we use to classify the Mannheimias that are going to infect cattle. The immunity to these are going to be it's going to be to that leukotoxin and the other cell antigens. So if you are using a Pasteurella vaccine, you want to make sure that they have a leukotoxin and a cell antigen component that'll help reduce some of the severity of these diseases. Our second culprit is going to be Pasteurella multocida. So this is a cousin of that Mannheimia. They're a little more broader in host range. You can find Pasteurellas in everything from your cat to rabbits to poultry. They are quite opportunistic, so they'll, anywhere there's compromised tissue, they're going to set up and cause a chronic infection. You can see the picture here. We got a lot of what's called fibrous and fibrin on top of the lung pleura. These are going to be a chronic pneumonia that comes after they've had maybe 1 or 2 other pathogens. So these are also isolated in conjunction with things like Pasteurella or Mycoplasma. You can see they have their clearance factor is going to be this capsule, so they have this real mucoid-looking colony. That's going to help them try to evade that immune response from coming in and inactivating it. Our third one is going to be Staphylococcus somni. Had some problems with this, especially this year. We saw quite a few cases. It's going to cause not only pneumonia, but you'll see vasculitis, meningitis, you'll get brainers, and then cardiac, you can get heart abscesses. Diphtheria, you can also get honkers from infections with this. These can be challenging to isolate in culture, so if you want to get a definitive diagnosis, they really need to be cultured within a day or two. They're going to require CO2 supplementation, and we use a special media. Here's what those look like on culture. You can see here's a brain that's had a Staphylococcus somni infection. There's a big abscess here, and that caused that's why this brain's on our necropsy floor. And then this is a lung that's had an H. somni pneumonia. So these are going to spread via blood, and they're going to multiply in the white blood cells. Others, we have Mycoplasma bovis, Pasteurella pyogenes, Biberstinia, and Salmonella. Pasteurella is another one we commonly see with these necrotic pneumonias that are long-term chronics. Mycoplasma bovis, we look like we have small microabscesses or little small caseous lesions, and then Salmonella can sometimes cause pneumonia in younger animals. So we wanted to see what the most prevalent pathogens are that we see in case submissions to the Veterinary Diagnostic Center. So I pulled a couple of years of data, and we looked at 1,243 cases that we got from feed yards on separate accessions and separate submissions. Our most prevalent one, like we thought was going to be, is Mannheimia, so we had about 300 of those, followed by Histophagus and Pasteurella, and then mixed growth or no pathogens. If the tissue's too rotten or it's been treated right before we get it, we often have trouble growing those organisms, so we do get some no growth. Here's what that looks like on a pie chart. You can see that Mannheimia is our most significant pathogen that we see in lungs, followed by Histophagus and Pasteurella, and these other minor, we may have a few handful of cases of E. coli or Salmonella pneumonia. So those are our most prevalent cases in organisms that we isolate with BRD. So now you know some of the bacteria that are involved. How are we going to treat it? So we're going to use an antimicrobial or an antibiotic. These classically were natural or synthetic chemicals. The first ones were metals like arsenic or mercury that they used to treat syphilis. So probably not a real good antibiotic that you'd want to be given. The next ones are natural or semi-natural chemicals that they found associated with molds. So things like penicillin molds were the first of the natural chemicals. These are secreted in nature by bacteria and fungi to try to kill their neighbors. So they want to establish a niche in the environment that they live in. They're going to secrete these antibiotics and try to kill their competition. So that's what role these play in nature. And us as veterinarians, we try to exploit these chemicals to treat BRD. There's two different types of antimicrobials. You may see ones that say they're bacteriostatic. So those are going to inhibit further growth, but they're not going to kill the bacteria that are already there. And then there's some that are bactericidal. So those are going to outright kill any bacteria that may be in that lung. They work by a lot of different mechanisms. So depending on the mechanism, you're going to have different ways that the bacteria have evolved to resist those antimicrobials. And this is dependent on the class and the composition of the antibiotic. The first one and the sort of the biggest group are the beta-lactams. These include the penicillin, so the classic drugs like penicillin, the semi-synthetic penicillins, and what are called cephalosporins. These mainly weaken the bacterial cell wall, so they're going to predispose that bacteria to environmental stress like salts and different ions. They're going to be more susceptible to the environment and the immune system, so they're more able to be killed. They have different efficacies of the groups of bacteria, and these are often called generations. So if you see a first, second, or third generation cephalosporin, that's about what types of bacteria those are going to target. So if you see those on a label, that's what that means. The ones that we use against BRD are going to include penicillin and ceftiofur. The trade names in these would be like Xceed, Xenel, Maxell. Those are all versions of ceftiofur. And the resistance to these are mediated via binding proteins, and I'll talk a little bit about what that might mean during an infection. Another group is going to be tetracyclines, so these are going to inhibit synthesis by binding to the ribosomes. Remember, those ribosomes are going to be the protein factories that the bacteria is going to use to synthesize more cell walls, more enzymes, more protein that they're going to use to metabolize and used to divide themselves. So these are going to accumulate into toxic levels within the bacteria. They're broad spectrum. There's fed and injectable formulations. The resistance to these is going to be via efflux pumps and protection genes. These are going to be like the core tetracycline, the oxytetracycline, the orimycin, and LA200. Then macrolides are the newest group. These are big molecules with these lactone rings. That's what makes them a macrolide. Our first one that we had available was Mycotil, and then Draxin we got a couple years ago. Now we got newer drugs such as Zactran and Zuprevo. These are all the macrolide class antibiotics. So these are the newest group of drugs we have to treat BRD. These are going to inactivate bacterial ribosomes similar to those tetracyclines. They're going to shut down that protein factory. The difference is these are long-acting, so some of these will have therapeutic levels of the lung for 2 to 3 weeks. So they're going to concentrate in that lung tissue and stay there for a long time. They do require rapidly dividing microbes, so when you do treat them, you're going to want to treat them when you got those microbes there and they're growing. And then they do accumulate within the cells, so it'll go from the blood to the tissue at a 20 times concentration. Quinolones are another class we have. These are going to block the DNA, so instead of the protein, they're going to target the blueprints that the bacteria is using to make those proteins. These are completely synthetic. These include enrofloxacin, danofloxacin, which is Baytril and A180. The thing to know, and Dee will probably hit on this later, but extra label use is prohibited in food animals, so you can only use those according to the label route and the treatment schedule that's on that label. And only enrofloxacin or Baytril has a label for control, so it's important to know that those are that's a legal directive that you can only use those on label. Okay, so there's some of our drugs. How is the bacteria evolving to fight these drugs, and how are they staying ahead of the game? Well, they've been doing this for millions of years, so it's not necessarily the advent of these new drugs or new therapies that we have that's causing the antibiotic resistance. So they've been living in close proximity to other bacteria that secrete these compounds for a long, long time. So there's a lot of naturally resistant occurring bacteria that are out there that have evolved these different mechanisms. Some of them will also, during long-term treatment, can accumulate mutations that make them resistant. They may grow in different patterns to try and avoid the antimicrobial compounds. Some have acquired those resistant genes from naturally occurring mutants. So bacteria are constantly looking for competitive advantages, so they're out sampling that environment, trying to find genes or mechanisms that they can exploit to try and give them a competitive advantage. So a lot of times this will come from exposure to insufficient concentrations of antimicrobials at the site of infection. This will be when you have a lot of damaged lung tissue, the pharmacokinetics of the drug, you're not able to establish a drug level that's appropriate. So that's one thing to consider. That antimicrobial is merely exerting a selective pressure. Like I would say, there's no free lunch for these bacteria. They have to make these proteins that confer resistance. Those resistance have an energy cost. They gotta get extra nutrients from the environment to make these different mechanisms. So that there's a competitive advantage to bacteria that don't have resistance. So unless there's a selective pressure like an antibiotic, they're not going to naturally want to have these things, generally speaking. So how does this occur? They occur in what's called a selective window. So in this diagram here, these little boxes are going to represent a bovine lung. The big dots are going to be resistant bacteria. The small dots are going to be susceptible bacteria. If we treat that lung with different levels, so it's called a minimum inhibitory concentration, that's going to be that minimum amount of antibiotic we need to kill the bacteria that are there. So if we go at a low therapeutic level, we see no change after treatment in either the amount of bacteria or the proportion of susceptible versus resistant. If we use one of those intermediate or subtherapeutic levels, we're going to see that we only get those bacteria that are highly susceptible. We're going to have a higher proportion of the bacteria of these big dots that are going to be resistant. Or if we treat it one that's an appropriate therapy that's of a duration and dose that's going to select all these bacteria, we're going to get a large proportion of those that are both resistant and susceptible, and we're going to be left with a small number of susceptible organisms. So that the selection for these resistant strains is going to be in B, in that intermediate or subtherapeutic level, where we may not be able to get enough drug at a high enough dose into the lung to cause resistance. So how does this work? So this diagram here, this is a cartoon of a bacteria. This is going to be a Mannheimia or Pasteurella in the bovine lung. You can think of these as sort of like a nightclub going on. They have a lot of metabolism. They're actively breaking down compounds. They're getting all those sugars and proteins from the bovine and trying to break them down. So they want to keep those antibiotics out. So how are they going to do it? Well, the first steps are going to try to keep it out. So they're going to have a security system that's going to try to prevent those antibiotics from getting in. So depending on the drug, some drugs can have more difficulty getting into bacteria than others. Bacteria have outer membranes, they have cell walls, peptidoglycan. So they have 3 different layers of walls to try and keep these out. So that's the first step. They try to prevent the antibiotic from even getting in. The second one is they're going to, once it gets in, they're going to pump it out right away. So they have these efflux pumps or bouncers that are going to kick that antibiotic out as soon as it gets into the cell before it can accumulate to a toxic level. So they're going to keep it out and then they're going to pump it out. Then if it is able to get in and they can't keep it out or get rid of it, what they do is they change the lock. So these antibiotics are sort of like a lock and key. They're going to try to unlock that protein factory and try to keep it from working. So they're going to mutate those locks. They're going to prevent the antibiotic from affecting the protein factory. Then the fourth way is they're just going to change the key. So if you think of that antibiotic like a key, they're just going to cut that key in half, and so it's not going to be able to have its effect. So they have 4 main mechanisms that they're going to use to try and reduce the impact that drug's going to have on the antibiotic. Okay, so specifically one of the trends we've seen emerging, at least in our lab isolates, is macrolide resistance. So in bacteria like Pasteurella, these resistance is conveyed via several mechanisms. So they have the whole package here. They have ways to change the lock. They have ways to bounce that drug out. They have ways to change the key. and often they'll have all these at once. So they're really geared up to be resistant to these macrolides. Another thing that they found is these are transported on what are called ice elements. So these are little gift boxes that the bacteria has evolved that can package up all these resistance mechanisms in one package, and then it can send it to its buddies. So it's gonna share these resistance mechanisms via different pieces of DNA that are easy for it to send to its friends. So some of the recent research shows that it can move up to 11 resistance mechanisms via ice elements, and that these can be shuttled between Pasteurella, Histophilus, and Mannheimia, so they can exchange these, at least in the lab. There's a lot of active research seeing how these are working in the bovine lung and what that means as far as resistance is concerned. So how do we test these in the lab? We do what's called phenotypic antimicrobial testing. So we'll do an in vitro or in the lab test to look at how sensitive these are to antibiotics. So the first step is we get a culture from a lung or a nasal swab. If you got an outbreak of BRD, we'll grow those up, isolate and identify, is it Pasteurella, is it Mannheimia, is it Haemophilus, and then we'll run these on what are called clinical laboratory standard protocols. So these are going to be a disc diffusion or a broth dilution. We do a broth dilution method, and that basically means we grow the bacteria in different dilutions of the antibiotic to see at what level the antibiotic can overcome the bacteria and vice versa. So here's what that looks like. So this is the disc diffusion method here. You can see we have a plate that we've streaked out with the bacteria all the way across it. And then we drop little paper discs that are impregnated with a specific concentration of the antibiotic. We are then able to measure this, what's called the zone of inhibition. So you can see these clear areas. That's where the antibiotic has killed the bacteria as it diffuses through the media. And we can measure that and say, okay, if this is 14 millimeters, this antibiotic it's going to be susceptible or resistant to this antibiotic, certain pathogen. We can also do it in a plate format. This is what we do in the lab. So here we have a tube of bacteria in a growth media, and we will inoculate a plate that contains a wide array of antimicrobials that you may want to use, and at different dilutions. So we can determine if the organism is sensitive, intermediate, or resistant to the different antimicrobials that you may want to use. So that's how we do it in the lab. Then we can interpret these. So the pharmacologists have done a really outstanding job with BRD and trying to figure out what do these lab results mean and how can we translate that into a real life situation that you're going to see on the feed yard. So but these aren't perfect, and I'll talk a little bit about that. What we can do is they have levels that they've established. They say if it grows at this dilution and with this drug and it's this organism isolated from this tissue, we can say it's sensitive, intermediate, or resistant based on those growth patterns. So initially we had to use a lot of human data because there wasn't much established for veterinary medicine. For BRD, we now have pretty good guidelines on the different bug-drug combinations. So I'll show you a little bit of our lab data. One thing you notice is this is data from the lab, and we don't really know how we can translate that to the lung because there's a lot of different variables. But I'll show you what we have. We have isolates from bovine lungs. We looked at Pasteurella and Histophilus from 2013. And then Mannheimia, I wanted to compare our data from 2013 with what we had from 2010 to 2011. So we looked at about 100 isolates each year and then compared what we were seeing as far as sensitivity patterns. If we look at Pasteurella, this is our data from last year. You can see we have at the bottom, these are all the drugs that you may want to use in a BRD outbreak. These are all drugs that are labeled and approved for treatment of BRD. And these are this is the percent susceptible. So 100% means that 100% of those isolates we got fell into the susceptible interpretive criteria. So all these bacteria in the lab were sensitive to these drugs. So you can see that ceftiofur and danafloxacin, we had a high degree of susceptibility. Almost all these drugs were pretty effective in the lab against Pasteurella. Oxytetracycline, we did see a little more resistance. Histophilus looks pretty similar. We have almost all the drugs are between that 80 and 90% susceptible range. except oxytetracycline, so that's maybe a little less effective. So everything looks pretty good. You have an 8 in 10 chance of having a susceptible isolate with most of these drugs. So here's Mannheimia. These are our isolates from 2010 to 2011. So you can see even 2 or 3 years ago, it looks like they're maybe a little more resistant than they are than those Pasteurellas and Histophilus. But we still have most of the drugs between 70 and 85% susceptible. If we look at our isolates from last year, you can see these go down quite a bit. So we're seeing in the 60 to 70% range for a lot of those drugs, except for ceftiofur. When we compare those, we see that we have between a 5 and 20% reduction in susceptibility for most of the drugs to Mannheimia. So we are seeing some changes in our resistance patterns that we get at the lab. You can see that here on the end, some of the macrolides have a little more than some of the others. So we're trying to see what that means and what genes might be involved in our lab isolates to determine what's going on there. There's a lot of bias in these results since this is lab data and we're talking about trying to see what clinical outcomes we may have in the yard. A lot of these isolates are from very sick animals that might have had several treatments or other issues. So they might have had 2, 3, 4 different treatments and that might have promoted some resistance. We don't know if the resistance of the antibiotic and the treatment failures are related. It might just be an incidental finding. So increased exposure or usage of macrolides. There are several new drugs available, so there's maybe more usage going on out there, and that's just related to increased use of this class of drugs. What we really don't know is if, and we're interested in finding out, is the increased prevalence of these resistant organisms is having some effect on clinical outcomes in the field. So is there more treatment failures because of the resistant phenotypes? There has been some data from other groups. This is from Ryan Lubers at Kansas State. He published a study looking at multi-drug resistance in Mannheimia, and they wanted to look at if these bacteria are resistant to these antibiotics, are they resistant to more than one? So they looked at this over 3 years' time, and you can see that this number 5 indicates 5 classes of antibiotics that it had resistance to. You can see in 2009 there was low prevalence of that. In 2010 they had almost 25% level, and then by 2011 it was at 35%. So there does appear to be some trends towards multiple resistance or multiple drug-resistant isolates in the Mannheimia. Okay, so this all goes back to success or failure treatment. What different factors is that going to have? What effect does these susceptibilities have on the outcomes in the yard? Well, the first thing is that we got to achieve a penetration of the antimicrobial at the site that's going to be comparable to what we're testing in the lab. So the pharmacokinetics of those drugs have a big factor, and depending on the status of the animal, if chronic, if that tissue is compromised in some way, that's really going to have a big effect on the drug levels that you can get at the site of infection. So that's the first consideration. The second is going to be is the drug is sufficiently potent and effective against the pathogen. So is that bacteria and that drug concentration, are they going to be able to, is that going to be sufficient to kill or slow down the bacteria? And then are we treating it long enough to allow a recovery? So all these things sort of come together to decide, is this going to be a successful treatment, or are we going to have a treatment failure? So the first one is that drug going to be able to penetrate the lung. This is going to depend a lot on the drug itself, the adequate blood supply. Is that lung, are we catching them early enough that there's still a good enough blood supply that we're going to be able to, the drug's going to be able to diffuse through the lung to the where the infection is occurring. So this is really going to depend on that timely treatment. So are we getting them pulled early enough? Are we getting them treated that it's going to be able to have an effect? Are we using the right drug? So if we have a Mannheimia or a mycoplasma, we might treat those infections a lot different. So if we're using Beta-lactams against something like mycoplasma, they're not going to be effective. Is the treatment duration, is it sufficient that the immune system can overcome? So it might take a week for that system to overcome and get into the recovery phase. And we want to minimize the time in that selection window. We want to make sure we're getting our drugs there and timely enough that we can minimize the amount that the bacteria are in that mutant selection or resistance acquisition window. So our take home is that treatment, that timing is going to be key. We want to try to catch these early when they're able to respond appropriately to the drugs. We want to avoid that window where they're going to be able to acquire resistance, or we're going to select for resistance. Our macrolide resistance in the lab appears to be increasing, but we don't know what that means as far as clinical outcomes in the field. And this is an active area of research that a lot of people are trying to determine. As you know, BRD is a complex problem, and there aren't any simple solutions, so we got to approach it from a variety of ways. And I'd encourage you to work with your veterinarian. You want to develop protocols that are going to be appropriate for your situation, and then follow those protocols and guidelines to really ensure that we're doing the best job we can at treating these infections. So, my lab's also in charge of the trichomoniasis reporting. So I want to make sure everybody's aware that we do have new trick regs, at least in Nebraska. So if there's a trick diagnosis as your bulls are going through breeding soundness exams, you need to report those to the Department of Ag within 5 days. And then they have some forms and neighbor notification. This is a new law in September. So I want to make everybody aware of that might not have heard. So this is the 800 number on the website. You can talk to the Department of Ag if you have any questions about that new legislation. For acknowledgements, I acknowledge Dr. Bruce Broderson. He's a pathologist that works on a lot of BRD, so a lot of the images are from him. Dr. Doster for supporting me and letting me come here and talk to you today. He's our director. And then my staff in the bacteriology and parasitology lab. And this is our new diagnostic center. We're starting the planning now, and hopefully by 2017 we'll have a new lab. So with that, I'll thank you, and if you have any questions.